All right, so guys, I'm going to go through a little bit of what we already talked about, and if there's some of this that we didn't have, you'll need to stop it. But I think if you were here, everybody should have all of this. So, cell cycle and cell steps, just to get us back up to speed from having had a break. Cell divides, then it has to grow and multiply those organelles that are necessary for its kind of general function, for it to get and utilize energy. Then it undergoes the S phase, and the S phase is when the DNA gets copied so that it can split again. And then G2, it makes the proteins that are necessary to split it again, and then it does in fact divide again. During interphase, so going back to this, most of the cell's lifetime is spent in interphase, but it's important to note that there's a lot going on there, even though it is interphase. And so in G1, it's growing and multiplying organelles. S, DNA is copying. G2, proteins needed for division are produced. Then specifically during the M phase, there are, again, two parts of that. Mitosis, which is division of the nuclear material or the DNA, and then cytokinesis, which is division of everything else. Together, they make two daughter cells. We always refer to them as daughter cells, okay? And those cells, as we've said a number of times already, should be identical to the parent cells. The only way that they wouldn't be identical is by way of an error or a mutation. Otherwise, if everything goes properly, they should be identical. And so mitosis might be a way of that living things have developed to properly pass on the correct information to each of those two new cells. Control. Stages have to follow one after the other. They have to divide at the right place and at the right time. And that's done by these kind of checkpoints, which again, it's not like somebody's watching the assembly line and shutting it down if something isn't right. These are chemical signals. If everything wasn't copied correctly from the previous generation of cells, G1 should shut it down and destroy that new cell because it doesn't have all the information it would need to proceed in a healthy way and an efficient way. Then it goes all the way around to where it's almost ready to divide. And if the DNA has not completely replicated, which it's supposed to do over here, and it gets ready to divide, it shouldn't continue there. It needs to finish replicating or copying. And then the M checkpoint is it's going to stop if the chromosomes don't line up. So here's what I told you before. Don't remember. So, for example, we as humans have 23 pairs of chromosomes, and they need to line up with homologous pairs. That is 1 and 1 and 2 and 2, right? Right down the line. Well, if they lined up like this with a 1 and a 2 here and a 2 and a 1 down there, then when they divide, one of them's going to get duplicate copies of 1 and one of them's going to get duplicate copies of 2, and they're not each going to have all the information they need. So the chromosomes need to properly align. Those are the checkpoints that we just talked about. Okay? And then here I think we're getting on toward some things that we haven't discussed yet. Cyclin is the chemical signal that makes it continue. Most likely acts at growth 1, after growth 1. And those signals, in other words, determining whether it's in the proper place for the next thing to happen, can either result in it pushing it ahead, that is release of cyclin to make it continue, or something called capsases, which would be an enzyme that causes the cell to be destroyed. And the importance of that is, if this cell is not properly formed, it's going to hurt the organism if that cell passes on that malformed form, I guess you would say. Okay, cancer, uncontrolled cell division. So now, if cells are cancerous or a tumor is cancerous, instead of it going to here and then having a check and then going to here and then having a check and then going to here and having a check, it's just going around and around in a very uncontrolled way. And the result of that is that it makes lots of cells, but not necessarily well-functioning cells. They're not density inhibited. In fact, probably some of you are familiar with the fact that a way that someone might know that they have a tumor is from discomfort. Let's say that, that tumor is pushing against their lungs or something, and so now they're kind of short of breath and have that discomfort. And it could be that this growth in their thoracic cage is actually growing out and reducing the volume of their lungs. Or maybe a more common one that many of you have heard of is when someone has a brain tumor. Sometimes it'll cause them loss of balance, sometimes it'll cause loss of consciousness, sometimes it'll cause severe headaches, sometimes it will cause impaired hearing or impaired speech, or even eyesight, because it's pushing on that part of the brain. Now, here's what I want you to be aware of on that, is that has to do with the fact that those cancer cells are not density inhibited. So you've probably never thought of this, but we're going to talk about it now. How does your brain know to get just the right size to just fit tightly inside your skull? Or how do your lungs know to get just large enough to fit just inside your thoracic cage? Well, the answer to that is they are density inhibited. That is, when they get large enough that they are pushing against a barrier like that, they stop growing larger. But cancer cells don't work like that. So now if we have a brain tumor, and this is the brain tumor, it's going to start to push, it's going to keep growing and expanding and creating pressure on other parts of the brain, and that's what triggers whatever it is that allows the person to know that something's wrong, whether it's loss of consciousness again, or headaches, or vision is affected, or whatever, is because that tumor is pushing against that part of their brain, and there's no room for it to expand. A fully developed skull can't get any larger, and so that's a problem with it not being density inhibited. And cancer cells don't have to be anchored. We didn't talk about this, but like in senior anatomy physiology, they're talking about how oftentimes tissues will have kind of a base layer, and that's what it hooks onto. But these don't, cancer cells can just kind of grow out in their own space, so they don't have to be hooked to anything. The brakes are off, and what we mean by that is that cell cycle is just going out of control. And cells from malignant tumors will spread. If they're malignant, so the other word is benign. So if the tumor is benign, it means it's kind of self-contained. It's not moving out. But to metastasize means to reach out with these kind of finger-like projections, and it reaches into other tissues. And so things like breast cancer are oftentimes metastatic, meaning that they reach out. Sometimes they aren't, and so then that's easier to treat. But anytime it metastasizes, it's much more difficult to treat once it's reached these little kind of finger-like projections into other tissues. And that's where we get into, if you've heard of somebody having cancer, well, it started in their esophagus, but then it spread to their lungs. Well, this tissue reaches out, these tumors reach out and affect other areas. Now, I'm going to probably go faster than what you're able to write. And so if I do, just have somebody stop the video, or if you're watching it on your own, stop the video and get caught up on the writing. But in the interest of getting through a little bit more material, I'm going to continue on. But if you need to, just stop it and finish writing and then continue on. Okay, some types of cancers. Carcinoma, from covering cells. And so skin or intestines oftentimes are affected by things called carcinoma. Sarcoma, okay, sarco is flesh, and So muscle or bone cancers would be called sarcomas. And then blood cancers or lymph cancers, which are kind of similar. You have sort of two different circulatory systems. You have the part that's carrying your red cells, and then also the part that's carrying fluids. Those kind of those things sometimes are together and sometimes not. But those that have to do with some kind of circulation are leukemia, lymphoma. Okay, so what do treatments for cancer do? Well, somehow it has to disrupt the cell cycle. So if we go back to here and we say that when cancer is happening, it's just going around and around. And none of these, it's just blowing through all these stop signs. And that causes all kinds of problems because it's making a lot of cells, but it's not making functional cells. So when we talk about treating cancer, we talk about how can we disrupt that kind of out of control division. So cancer cells are dividing more rapidly, so they're more affected. And so radiation knocks back a tumor. We know that radiation can damage our tissues. We know that because when we go in even for X-ray radiation, which is diagnostic, and we need that to tell if we have something going on with our skeletal system. But as you well know, when you go to the dentist or to the doctor and have an X-ray, they're very careful to kind of isolate that X-ray to the one spot that they're trying to find something out about, because they don't want to expose all of your body to that radiation because it can be damaging. But in this case, when radiation is being applied to treat cancer, that is the intention, is to damage the bad tissue, is to knock back the bad tissue. And then chemotherapy drugs, you're probably familiar with chemotherapy drugs, but those disrupt the cell division. So we said that cancer was uncontrolled or too rapid and inefficient of cell division, and so these are meant to disrupt that cell division. Now the problem with either of those is how do we damage as little tissue around the cancerous area as possible? And how do we only disrupt mitosis in the cancer cells and not as much in the healthy cells? Now this bullet point up here kind of tells you about that. When doctors are treating these things, it's going to have a more marked effect on the cancer cells because the cancer cells are dividing more rapidly. So your healthy cells aren't dividing as rapidly, and so that, although that may be disrupted, it may not have a really detrimental effect, although usually there are some side effects. If you know people who have undergone cancer treatments, chemotherapy, they'll really, doctors will really go at it aggressively if they have an aggressive type of cancer. And then once they feel like they've kind of knocked back the cancer and kind of challenged the person's healthy cells too much, then they'll stop for a while and then see how the cancer is progressing and then come back another time and so on. So you may have heard people talk about they're going through their second or third round of chemotherapy or whatever, because two things, I think, they can't continually give it to people because it makes them, the side effects are too significant. And the other thing is they need to kind of take a break and then reassess what kind of condition the cancer cells are in. Are they knocking them back? Are they having the desired effect, and so on? So some of the things that are used, amitotics. So mitosis means to divide, for cells to divide. Amitotics are meant to keep it from dividing. So anti or amitotics. Vinblastine prevents the spindle from forming, and Taxol freezes the spindle, doesn't allow it to be effective. So those are just some examples of things that might be administered to someone who's undergoing cancer treatment. Then something that holds some promise are molecular therapies. And the goal of those molecular therapies are to actually fix the genes that code for that particular protein. So that you may have heard of or heard referred to as gene therapy. And gene therapy means you're actually trying to fix the mutation that's causing that irregular growth or cancerous tissue. Okay, so now we're going to get into the particulars of mitosis, and I think it would be helpful if you had your book open to page 85. So I would open your book to page 85, and I'll probably put a diagram or two up here as we go through this so we can talk about it a bit more. But mitosis is the division of chromosomes in such a way that each of the new cells has the same number as the parent cell had. So you take, in the case of humans, our body cells, we have 46 chromosomes or 23 pairs of chromosomes. Mitosis in our body cells would involve duplicating those 46 chromosomes and then dividing them so that each new cell has a full complement of 46 chromosomes. What's called the diploid number, diploid meaning both of each pair. And a chromosome is a rod-like structure that's seen during meiosis and mitosis, excuse me. It's DNA that's folded and packed around histones or proteins. So important to note that chromosomes are actually made up of two different parts, proteins and DNA. So you might think of the important part of it's the DNA. The proteins kind of hold it together. But later in the year, we'll talk about how scientists were able to discover they knew that these chromosomes were made of these two parts. One of them was simpler, DNA, and one of them was more complex, proteins. And of course, knowing how complex an organism is, scientists kind of assumed that the proteins were the part that were giving the message. But as it turns out, they did some kind of clever experiments and figured out that it was in fact the DNA that's passing on that information. Okay, so let's look at chromatin and chromosomes. So chromatin would be kind of like this, just this kind of a loosely bound piece of string. There's this singular strand of DNA and proteins that it's sort of, you might say, elongated. And so that's when the cell isn't dividing or when the cell's in what's called interphase, this is what the DNA is going to look like, just this kind of loosely bound combination of thread-like material, thin threads and proteins. Now when a cell gets ready to divide, it needs to kind of compact that, and so that loose combination of thread gets kind of coiled and rolled and looped and condensed to make something called a chromosome. Now, we talked about briefly the other day, the reason that that's important is if you have this loose thing and you just kind of haphazardly cut it in half, it would be hard to think that we would get the right portion in each new daughter cell. But when we have it in this condensed form, and this actually duplicates, right? So we have two of these chromosomes, and then they pull apart, which we'll look at in a little bit. Now we know this is a complete set, and this is a complete set. So that folding and coiling into a chromosome ensures that each new cell gets a full set or complement of information or DNA. So this picture at the bottom is it's basically a layout of a set of human chromosomes. And as I told you, humans have 23 pairs of chromosomes. So this is pair number 1, pair number 2. You can see how it's numbered down below here. All the way through 22. And then the other one's not called 23, it's called XY. And the reason for that is these are considered two different things. So So these are the sex chromosomes, and all the rest of these are the non-sex chromosomes. And by sex chromosomes, we mean the ones that determine gender. So the gender ones, and we actually know from looking at this set of chromosomes that this was a boy, this was a male. And the reason we know that is this person has an X chromosome and a Y. Now, something else I want to point out to you is up here with the asterisk, it says each chromosome has a matching chromosome. Same size, same pattern, same centromere position, genes control for the same traits. So if we look at all these, do you see that not only are they number ones are the same length, but the shading and the lines in them are all the same. Same length, same shading and lines, same length, same shading and lines, same chromosome, which is where they're kind of pinched together here, and these look alike and these look alike. They're all paired. It's not done randomly. Now, it says matching, but not identical. The critical part of this that you need to understand right now is, as an example, this child got this chromosome from mom, and they got this chromosome from dad. And let's say that this chromosome carries eye color, hair color, and blood type. Eye color, hair color, and blood type. So they both have to carry those characteristics, eye color, hair color, blood type. But this one from mom might be blue eyes, blonde hair, and type A blood. And this one from dad might be brown eyes and brown hair and type O blood. Do you follow what we're saying? They code for the same trait, but they don't code for the same form of the same trait. Right? That's a really critical thing for you to understand. So these two chromosomes are called homologous. They're homologs. They're not identical. They're not copies of the same thing, but they are for the same traits. Okay? And that's true of all of these. Now that's where this one is different. In males, males have one X chromosome and one Y chromosome, which, as you can see, is only about half the size. Okay? Females have two X chromosomes, so they actually have 23 sets of homologs. Boys have 22 sets of homologs and one set that's not homologous, because this Y doesn't have everything that the X has. Now, it has some of the same traits, but some it just doesn't have at all. And that's one of the reasons that you end up with some traits or conditions that are more prevalent in boys. And one of those, as you may be aware, is color blindness. Okay? Anybody in here know anybody that's colorblind? Raise your hand if you know somebody that's colorblind. Okay. Now, if you do know somebody, share with them if that's a man or a woman. And in almost all cases, that's going to be a man or a boy. And so I'll give you an example from our school. I think maybe a couple of the Davies brothers might be colorblind. And so it's something that happens. And if you remember Mr. Elsasser, I think he was colorblind. And it's not that uncommon, but it's very uncommon for a woman. And the reason is, we won't get into it too much because we'll talk about it a lot more in genetics. But for a boy to be colorblind, they only have to have one colorblind allele. But for a girl to be colorblind, they have to have two, because colorblindness is what's called recessive. So if you have two alleles and one of them is good, you might say, your ability to see color is going to be fine. But if you're a boy, you only have one, and it's for colorblindness, you're going to be colorblind. Okay, I spend too much more time on that, but I think it's worth noting a difference there. In humans, this is 23 pairs of 46 total chromosomes. Okay, so haploid means one of the chromosome pair, or one in or just in. So if we go back to our diagram and we said, this is not true, but let's say that the one on the left came from mom. So this one, and this one, and this one, and this one, and this one. So if we said we're only talking about the ones that came from mom, that would be haploid. But when we add to it the ones that came from dad, 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 now it's diploid. Okay? So haploid is just one of the chromosome pair, one N or just N. The only thing that humans have, the only cells that humans have that are haploid are gametes. So with gametes, we're talking about eggs and sperm. All of our other cells have 46 chromosomes, but eggs and sperm only have 23. I think with one of the classes the other day, we talked about why that's significant. Well, if the offspring is going to have 46, then it can only get 23 from dad and 23 from mom to have 46. If it got 46 from mom and 46 from dad, it'd have 92. And the next generation got 92 and 92, it'd have 184 and 368 and 736 and so on, right? So it's really vital that we have in meiosis this reduction division. But that's the next topic in the chapter. Right now we're going to really focus on mitosis. So here's what that looks like. When we have just a healthy cell, each one of the chromosomes, the one from mom and the one from dad, just look like this, just a single strand. Each chromosome is just a single strand. Okay? But when it gets ready to divide, it's going to duplicate. So now the one from mom looks like this, okay, like my knuckles are connected and my two index fingers are identical. These now are identical. And then the one from dad does the same thing, and it's identical. So what you really end up having is you have, let's see, let's go back to this diagram again. So this one would be two strands, and this one would be two strands, and two strands, and two strands. Okay? And so then that allows for, when it divides, these centromeres divide, and now this, here we're only talking about eye color, hair color, blood type from one parent. So let's say this was mom. Then mom's doubled. So we said she was blue and blonde and A. I'm going by memory, but we'll say she was blue and blonde and A. So this one is also going to be blue and blonde and A because it's an exact copy. This is not a homolog. This is an exact copy. Then when it divides, each of the two new cells that mom has herself are going to be blonde or blue, blonde, A. Blue, blonde, A. Okay? See if I can get that. I don't know what we got going on there. If I can get that out of there. Maybe not. Okay, there we go. All right, so let's move this back up so we can see the whole thing. Right. All right, so DNA replicates before mitosis. This is talking about mitosis. The DNA replicates. That means it goes from one strand to two strands, two identical strands. A duplicated chromosome consists of two sister chromatids. Sister chromatid, sister chromatid. During mitosis, the centromeres divide. That's this white chunk here. And each sister chromatid becomes a daughter chromosome. Sister chromatid, now they divide, daughter chromosome. Sister chromatid, now they divide, daughter chromosome. Each daughter gets a complete set of daughter chromosomes. So again, we're going back to this. Blue, blonde, A, blue, blonde, A, blue, blonde, A. Two new cells, blue, blonde, A, blue, blonde, A. Okay, so in mitosis, a 2N parent makes two 2N daughter cells. So if we're talking about you getting a little scratch on your skin or something, and your body's making new skin cells, the old one had 46 chromosomes, and the new ones are going to have 46 chromosomes. And that's true of all your cells with the exception of sperm and egg. That's it. Those are the only ones that aren't 2N to 2N. A 1N parent makes a cell that undergoes mitosis and makes two 1N daughter cells. Okay? And so that actually happens in the making of sperm and eggs, but again, we'll talk about that more later. So whatever it is, this is an important idea right here. Mitosis maintains chromosome number. Mitosis maintains chromosome number. Can't see that very well. Let's try that. Okay, I don't know if you can see the color difference very well, but mitosis maintains chromosome number. Whatever it was, that's what it's going to be. 1N to 1N, 2N to 2N. There's no such thing as 3N, but whatever it is, whatever it started with, that's exactly what you're going to have now. Okay, then the next thing is stages of mitosis. But guys, I think we covered enough with that today. I know if you were able to keep up writing, you're going to still have a little bit of time. But what I would like for you to do is continue the questions. You were to have already answered through 16, and so I'm going to have you continue through 29. So continue the questions through 29.